Okay, I think we can get started. Hello, everyone. Welcome to this special lecture of the NCI-funded Big Data Training for Cancer Research workshop. My name is Min Zhang. I'm Professor of Statistics and Associate Director of Data Science at Purdue University Center for Cancer Research. It's my great pleasure to introduce our speaker for today, Dr. Sean Davis. In order for Dr. Davis to have the most time with you, I'm going to keep my introduction brief and share just a few highlights. Dr. Davis got his MD and PhD after a bachelor degree in mechanical and aerospace engineering from Purdue University. From Princeton University. He completed his residency in pediatrics and then fellowship in pediatric oncology. So Dr. Davis worked at NIH, mostly NCI, for 16 years before moving to University of Colorado last year. His research interests are at the intersection of public data resources, software development, and cloud technologies. So with his wide range of expertise, so he gave a talk on different topics each year since we started to work together on this big data workshop back in 2016. So please join me in welcoming Dr. Davis.
All right. I'm going to share one more time the slides. So thanks to Min and certainly to John for being here and organizing. And I've known Nadia as well for quite some time. It's a great team to work with. They're so clear in communicating, and they've really put together pretty much a pretty long history of data science education efforts and made a great transition to the virtual world. Not everyone's done that successfully, so I appreciate being involved here. I want to spend the next hour and a half talking a little bit about Bioconductor. As you'll see in the first few slides, an hour and a half to talk about Bioconductor is kind of a silly thing to think about because it's such a at this point a very large project with a lot of sub-projects associated with it and a long history. So what I say here is going to be my 2 cents worth, but I'd encourage you, folks who are thinking about spending time and energy with in biological data analysis to spend an inordinate amount of time looking at Bioconductor. As Min pointed out, I'm at the University of Colorado now. I wear a few different hats, but one of the ones that I've worn for the longest actually is my involvement with Bioconductor, and it's been a mix of formal and informal, and that's actually true of a lot of folks who've been involved with Bioconductor. It's a largely a volunteer effort, despite the fact that it's had really successful NIH funding for quite some time. The slides, the link to the slides is in the chat, and for those who might have missed it, bottom right of this first slide, or the QR code at the top left. And there can be a couple of little exercises as we go through, just to make this slightly more interactive. Although I'm not really great at Zoom interaction, but if you have the slides in front of you, it'll be easier to follow along. So I'm going to start out with maybe a little bit of a soapbox, but reproducible research, and particularly computational reproducible research, can be really challenging to do. And even when you do it, even when you have a reproducible analysis, it can still be wrong. And this is an opinion piece from Jeff Leake and Roger Peng from a few years ago. I'd encourage you to kind of look at it. But they make a couple of points in here that I'm going to highlight here. Reproducibility, or the ability to recompute results, and replicability are two foundational characteristics of successful scientific research. Some would say that there's been a crisis of confidence, and I'm hoping that crisis is being averted slowly. But we really, as a scientific community, must ensure that reproducibility and replicability are met to the greatest extent possible. And to do that, we have to take an approach that greatly expands data analysis education, which is a perfect forum to talk about, and routinely employs software tools. So why do I mention software tools? Well, software encodes knowledge is a take-home message. Bioconductor sort of is an encapsulation of that idea. But the concept is that if I can write software that does something useful to me, and I can document it in a way that makes it useful to someone else, then the knowledge that I have has been encoded in that software. And for those who think and talk about software, or think about programming languages, R is a programming language that really espouses the concept of very high level software tools. So what do I mean by that? Much of the software that we're going to be talking about today, or that I'm going to be implying, is in Bioconductor, incorporates a huge amount of knowledge into simple functions. And that's pretty cool, because what we end up with as Bioconductor users is the sum knowledge of all the developers and all the scientific work that they have built on to develop their software packages, and then the documentation that goes with it. So what is Bioconductor? Well, it's a large NIH-funded open source software community, and it's dedicated to the analysis and comprehension of high-throughput biological data. This used to read comprehension of microarray data, and then it read comprehension of genomic data, and now has really branched out to high-throughput biological data, all different types. Bioconductor by the numbers. The project started in 2002. There's a slide that I didn't include here that Vince Carey put together that includes the first email to the Bioconductor email list back in, I believe it was late 2001. It's kind of the beginning of the project in a sense. And it's been running continuously since then with varying NIH funding sources, various other funding sources as well. But it's been growing quite significantly. It's built on and in R. It's an open source software package, software platform for data science. R has between 2 and 3 million users worldwide, just an estimate. The numbers from 3 or 4 years ago, 15,000 contributed software libraries, kind of like plugins for R. And that's not including Bioconductor, it's just the general R community. And R is kind of cool in that it runs on the laptops to the largest computer systems in the world, installed and used quite regularly. Bioconductor enjoys a pretty big user community. It sees more than 50,000 unique downloads per month. You can look at the stats, you can follow the stats pretty easily. The Bioconductor team keeps track of those downloads and publishes those numbers. Conservatively, if you just look at publications that are easy to find associated with more popular packages, Bioconductor has over 22,000 PubMed Central citations. Bioconductor is released twice a year. And by released, I mean that all the software packages, data packages, etc., go through a formal testing process. And that testing process occurs on a specific version of R. Bioconductor version 3.15 was just released with over 2,000 individual biomedical and omic data science software packages. We'll talk a little bit more about experiment data packages and annotation packages. And in addition, Bioconductor really kind of espouses the concept of reproducible research and community engagement and education. So there are 29 workflows. These are really more complex documentation packages that encapsulate biological workflows. And then there have been 8 literate programming books. And by literate program, I mean that the text of the book, the code associated with that text, and the output of that code are all built into these reproducible books. Changes to the code or changes to text result in changes to the book. So they're kind of living documents. The Bioconductor project receives submissions of about 3 to 6 new packages per week and has hundreds of active developers at this point. And we'll talk a little bit about submission in a bit. This is the point where I pause and let you guys read all of the Bioconductor packages. And this isn't actually all of them. I think it's about 700 that fit on the page. That was a joke that you're supposed to laugh at, but obviously it's a little less impact, at least for the speaker here. So I want to talk a few minutes about Bioconductor capabilities. And I'm not going to spend too much time delving into individual packages or sort of how to use Bioconductor at this point. We'll do that in the second half of the talk. In general, Bioconductor supports all phases of the biomedical data science workflow, everything from specialized data import and export, data management. I think you guys had a discussion yesterday about data management and reporting. Bioconductor is built on the fair data principles. I'm not going to talk about those specifically, but really the Bioconductor capabilities and publishing, package publishing predates fair data principles, but largely Bioconductor meets those principles. Data integration and interpretation, including access to millions of public data sets. We'll focus a bit on that later. Context-specific analyses and statistics, visualization and publication, quality plotting, efficiency and reproducibility, even at scale. And reproducibility here, a lot of folks think about bioinformatics. Many folks think about bioinformatics as kind of running a set of pipelines or workflows. And Bioconductor sort of sits in this place where it can do all of that, but it also incorporates the ability to keep the human in the loop. That is, to analyze data interactively, to explore data. And a large part of what Bioconductor can do for you is to help you, guide you in data exploration and integration. And then finally, reproducible literate reporting. So Bioconductor quickly, very early on, adopted literate programming as an approach to reporting and also to documentation. And for those who haven't thought about that much, I'm sure you're going to hear about R Markdown, and that's really what I'm talking about here. Feel free, as we're going along, if you have questions, to drop them in the Q&A. I'll try to keep glancing over, and Min and Nadia can remind me if I'm missing something. So feel free to interrupt. Qualities of Bioconductor. Bioconductor is very discoverable. There's a website. It's updated quite regularly and automatically, based on a lot of the automation tools that Bioconductor uses to build reliable software. All pieces of Bioconductor are installable, and what does that mean? The software that you see available through the Bioconductor website or web is typically something that you can get access to. It's open source. That is, you can read the code, and you can install it into R, assuming that you have a version of R that's supported by Bioconductor, and vice versa. Every landing page, and you'll have a chance to look at a landing page in a bit, includes installation instructions. And if you have R running, then you should expect that most of these packages will install without a lot of fuss. Bioconductor is built to be quite reliable. So unlike most other, actually probably all other software projects that I can think of, large open source software projects like this, the entire project is built and tested quite regularly. And by quite regularly, I mean every day. So all the packages that are included in Bioconductor, over 2,000 of them, are built daily. And then we as developers can go to one of these pages that shows the results of that build process and lets us know whether our software is working correctly or is broken in some way. This also means that software that's broken doesn't end up getting incorporated into Bioconductor. So if I make a change to my software package and it for some reason breaks things, then the testing process, the building and testing process will catch that, and that new software won't be pushed into Bioconductor. We'll keep the old version that's working. The Bioconductor requires really for some submitters and developers to build and maintain documentation. And this documentation comes in a couple different forms. One is at the level of code. So as a fellow developer or a high-end user of a software package, I can look at the individual functions, their inputs, their outputs, etc. But also, because we have a biological audience, the software documentation needs to include the so-called vignettes. Each software package has sort of long-form documentation that describes how to use the package, potentially background knowledge, statistical models, and choices of algorithms, etc. Some of these vignettes can be hundreds of pages long, so they're really more like books or treatises, and some are shorter. The Bioconductor has extensive support built in at this point. Originally, there was an email list that was the way that we got support. And just to share how I got involved with Bioconductor, when I arrived in my lab back in 2002, we didn't have software tools for working with, at that point, microarray data. I looked around and found this new project called Bioconductor. The way that you interacted with it was through an email list. I started reading the email list. Then I started answering a few questions. Then I started answering a lot of questions. And that became how I became engaged with the project. We've now moved to an online question and answer forum. And for those of you who are thinking about engaging with bioinformatics in the future, I'd encourage you to sign up. And the question and answer forums here are very gentle. People tend to be very helpful and not make a lot of assumptions about people's background knowledge. So it's also a good place to learn through either reading questions or, like I said, answering questions can be really helpful. Bioconductor is really integrated. So unlike a lot of things, a lot of software packages that are purpose-built for high throughput data analysis in the Python world or even in C or C++, other languages essentially, Bioconductor is built to be fully integrated. So here you can see, for example, how the different pieces of RNA-seq analyses are encapsulated. So in blue are the various pieces of or the various formats of data, kinds of data that are involved in analyzing such experiments. And then in the pink are software packages that deal with those kinds of data. And the lines here between these different blue boxes are not only connections, logical connections about how to go from CSV counts to differential expression, but also their relationships between the packages in pink with the packages in the next pink box. So those lines represent real interconnections between software packages. And keep in mind that each of these software packages is typically developed by a different individual or a different set of individuals. So it's no small feat to have this integration. And like I said, in other languages, this isn't typically the case. Bioconductor is really connected with the community. So in the middle here is the Bioconductor, logo, and all of the things, all of the other boxes here are other large scale, in a few cases, cancer related data resources. And Bioconductor, when I say it's interconnected or connected, I mean that there are actual software tools that directly connect Bioconductor and Bioconductor users to data resources in these various other data domains or repositories. Come back to that in a bit. Scalable, Bioconductor can run analyses from the smallest CSV files to hundreds or even thousands of whole genomes, and there's software tools to make that possible. State of the art. Many of the software packages in Bioconductor are either one of a kind, that is, they do the best analyses, provably the best analyses of a certain type of data, or they stand as the sort of gold standard that people then use to reproduce that software, that approach, that algorithm outside of Bioconductor. So things like, in this case, we're looking at differential expression analysis of RNA-seq. There are a number of packages in Bioconductor that are really kind of state of the art, and multiple of them, so you can choose which one you want to try. And Bioconductor is driven largely by the community. So the grant support does not really, it's not enough to support all of the software development efforts that are going on in Bioconductor. Bioconductor is kind of the core. The grant support is for the core Bioconductor packages, and also for the infrastructure related to support the website, building and testing packages. But at this point, the Bioconductor grows mostly by people contributing new software packages or updating the software packages that exist with new capabilities.
Hey, Sean, there is one question I don't know
Yep.
if you want to answer now or later. So it's from Junhong Song. Yeah. So, Junhong, you can ask your question now.
Yep. I think you're muted, Junhong.
Uh,
Oh, sorry.
No, it's okay.
No, it was a mistake. I mistakenly pressed the button. Sorry.
All right, so we'll go ahead then. I'm going to take a quick break and let you guys do this little quick exercise. I wanted you to navigate to the Bioconductor website and then locate the list of software packages. It's on the website. It's under Install, and then Discover Software Packages. Use the search box or categories to find a package that interests you. And then once you find a package that you kind of like, it doesn't really matter which one, to be honest. Just pick one and examine the landing page. And the reason I wanted you to do this is that for those of you who've used Bioconductor, ignore all of this. But for those who are new, I wanted you to get a sense of the kind of information that's on the landing page. The rank of the package is useful for looking at sort of popularity. Not to say better, but more popular packages tend to be are more used. How long has the package been in Bioconductor? That can give you an idea of sort of how long the package has been maintained. How do you install the package? Just find it. You don't need to install it. And then finally, the link to an HTML or PDF documentation, the vignette. So I'm going to pause for just a couple of minutes and let people go through that. If there are any other questions, now's a good time to ask. I see a question in the Q&A. Bioconductor is a consulting center for how to use each package, including debugging. Not sure exactly what the question, exactly how to interpret the question, but Bioconductor isn't really a consulting center. It's a software project. The consulting piece could come in with using, when you think about the support site, or we'll come to Slack, the Slack Bioconductor Slack in a bit. So you can ask questions there about how to perform analyses, what packages might be available or useful. In terms of debugging, really the support site is a great place to ask questions. And for those who haven't asked a question before of a support site, having a nice reproducible example that shows how to create the bug or the error is really helpful. From Serendipity, does it matter that some packages are just example data sets, or should we select other types of packages for this exercise? I don't really. I don't really care. I wanted you to get to the website and look at it. Otherwise, it's very easy to get through a talk like this and not, for example, have bioconductor.org in your search history. Yeah, so you pick the package that you like, and the fact that you found an example data set means that you've stumbled onto different kinds of packages in Bioconductor. We'll talk a bit about those as well. Can Bioconductor packages be run in places other than R? That's a great question. It depends a little bit on what you mean by in places other than R. If you mean that can R packages be used in some place other than R running on your laptop? Absolutely. There are a number of different websites that perform analysis using Bioconductor packages, but R is running on somebody else's machine with a website in front of it, and the analyses are actually running in R using Bioconductor packages, but you're not the person sitting there typing the code to actually run the analyses. And in fact, since Bioconductor is open source and in many cases licensed in a way that can be used pretty broadly, there are some commercial companies that have used Bioconductor packages directly in their software for analysis. Are there any online courses that teach Bioconductor in addition to vignettes, and how would you recommend teaching it virtually? And Amy follows up with teaching Bioconductor packages, on a specific NGS topic. I'll come back to that in a bit when we talk about engaging, but we have a couple of different approaches. One is online course materials, and the other is a relatively new development called Orchestra that allows people to run RStudio and R in the cloud using pre-installed software developed around the idea of being able to teach Bioconductor in virtual environments, or in an environment where you don't want to have everybody have to install software. All right, great question, thanks. I'm going to keep going. So just here, I just wanted to show you an example of a package landing page. For those who got to one, this will be no surprise. In Bioconductor 4 years, and rank 330 out of 2140. You might say, oh, well, that's actually not a very high rank, but keep in mind that some of these packages are relatively specialized. So rank doesn't tell you everything. Build is okay. Dependencies, installation instructions down at the bottom. There's often a citation, etc. So a lot of information on the landing page, and this is updated daily, essentially. Switching back a little bit to conceptual kinds of things, a core value this is where I started. A core value of Bioconductor is reproducible research. And the first bullet point there is recognize complexity in high throughput biological data. And I used to be able to talk about these kinds of things pretty easily because they were front and center in people's minds, but there were some examples of real tragedies from a data science perspective, where some clinical studies, clinical trials were launched basically based on faulty analyses of at that point microarray data, and at least in one case, the fault in the analysis was due to a cut and paste error in Excel. And I don't want to poo-poo Excel. Excel's great, but these data, in order to encapsulate biological data sets that are very complex, Bioconductor has settled on this concept of a shared data container. And the data container that we're talking about is on the right-hand side. There's sort of three main pieces when we think about describing biological data. There's typically features. So these would be things like genes or chromosome locations or proteins. So we need information about what we're measuring, not the measurements themselves yet, just information about what we're measuring, and that's the data frame, or the matrix on the left-hand side of that figure, feature information. Then typically there's also sample information. And in Excel, sometimes people will include, for example, the name of the sample. But in pretty much all biological experiments, those samples have attributes associated with them. And yes, we can keep those attributes in a lab notebook, or we can keep them in a separate Excel spreadsheet and do analyses by pulling everything together. But the better approach is to attach that sample information to the sample names, etc. So that becomes another data frame or another matrix in the top right. Then there are the measurements themselves for each sample, for each feature. So let's use genes, for example. For each sample, for each gene, we have a measurement that becomes one of the blocks in our feature information, our assay data, we call it, in our assay data. So now we have, and that we tie all that together. Bioconductor ties all that together into a single data object. So when you, for example, when you take just a subset of the samples, you also get all the feature information, all the gene information associated with that sample. And if there happen to be more than one measurement, then you get all of the measurements associated with those samples. And the same thing you can do with genes. You can slice so that you're looking at just a subset of the genes. But all this happens in a way that everything's tied together. So there's not a really an easy way of doing a cut and paste error, because the sample information, the gene information, and the actual measurements are all one object. And because this object is understood, it's used throughout Bioconductor, I can write software that ingests information from a biological experiment or set of biological experiments. If I can get it into a summarized experiment at one of these data objects, then all the downstream analyses that might rely on the format of the data can use the summarized experiment as the format for those data. So it becomes sort of a single sort of communication, way of communicating between software input or data input, data analysis, and even in the case of data output, we can use some of the same data containers. And then finally, I mentioned fair data principles. In the bottom right, we can attach essentially arbitrary information to this data set. So who's the data owner? What is the project ID? Things like experimental conditions or approaches, standard operating procedures, even batches, which batches of things were used, batches of solutions were used, etc. So all that stuff can be attached to our data set, and it flows along without any extra work for us. Another piece of reproducible research is version control. So version control of data and also version control of software and code. Continuous testing and integration. And turns out that if you have an analysis that you need to rerun or rerun on a regular basis, you can actually use a lot of the same tools that we use for software development to rerun your analysis when something changes. A lot of people would think of a text-based workflow. That is, there's no real GUI, there's no graphical user interface for using R. A lot of people would think of that as a shortcoming. But because everything is text-based, you have to type code in order to make R and Bioconductor do something. You can save it. And once you can save it, you can version control it. So essentially, just by having a text-based workflow. If you're able to save if you do save all that information, you have a reproducible workflow, at least to a certain extent. Literate programming, we're not going to touch more on, other than just mention that that's our markdown we're talking about. Education on tooling and how to use them in a reproducible research environment, and numerous mechanisms for fair data sharing. And serendipity, I think, was the question, asked the question about data packages. So one way of really quickly and easily sharing biological data is to load those data into one of these summarized experiments in the context of a bioconductor, of an R package, and then share that R package. That R package has built into it authorship, version control, testing potentially. So once you share that with data inside it, you really shared the data themselves, but you've also shared a lot of metadata about the analysis and potentially even the whole analysis itself. So just to make it a little bit more concrete, if we think about this data structure, this summarized experiment that we just talked about on the last page, in the rectangle in the center, all of the analyses that are around the outside, and summarizing those through graphics, are immediately available from this summarized experiment. And that implies a few things. One is that the interoperability between software packages, and remember, these are typically built by different developers. The interoperability is through these data objects. And it also implies that some packages that are purpose-built for one type of data, for example, in the top right, we're looking at principal components analysis. If we can do principal components analysis, say on gene expression data, starting from a summarized experiment, we could potentially expect that if we had similar data, but proteomics or metabolomics or microbiome data even, if we can load it into a summarized experiment, we can probably run that same software package that plots principal components using without really a lot of change. Obviously, you need to make sure assumptions are met in doing so, but technically, that's what I mean by interoperability. Software packages that were built for one thing can be reused for another, and data packages or data structures that were built that are built to be general then allow us to interoperate between software packages. I mentioned that Bioconductor is really driven by contribution, and contribution happens at lots of different levels, but package submission is sort of one of the mainstays of how Bioconductor continues to grow. There's a fairly well-organized package submission process. Anyone's encouraged to do so, and the package submission process is really built to meet the developer where he or she is. So if you're submitting your first software package, you're going to get, this is all done through GitHub. You're going to get a lot of feedback or a technical review, and potentially lots of changes, and potentially help with those changes. For those of us who have done a fair amount of R programming, that package submission process can be fairly quick, and is a way of, and a lot of us use it as a way to just make the kinds of software that we would otherwise kind of keep private more available, just because we recognize that a lot of folks might find our software useful. The package submission process does not include a scientific review specifically. We don't really spend a lot of time thinking about whether a package belongs in Bioconductor, other than to say if it meets technical specifications of basically along the lines of data reuse and reproducible research, then it will likely end up being accepted. The contributions they're tracked through GitHub and done in public, essentially. So it's an open review. This only goes up to 2018, but this is the number of Bioconductor contributions. The blue line is an actual exponential fit. The Bioconductor project, at least through 2018, was growing exponentially. And I can't say for sure, but I wouldn't be surprised if that continues. We have partnerships with publishers or other groups to help publish our work, but also to help build community. So F1000 Research is an online post-publication peer review journal, and they've partnered with us to establish basically like a sort of subsection of their journal that's Bioconductor related. This is kind of nice because it can be a little challenging to publish software packages if the reviewers don't understand software development. So in this context, we can ask for reviewers to be developers. That is, we can suggest developers as reviewers, and reviewers understand that what they're reviewing is a software package. So it's kind of a nice approach to making sure that we get credit for all our work. I mentioned Bioconductor is pretty connected, same slide as before. And then this is a slightly different slide. What's shown here is our cloud and very large scale data platforms, including Google Cloud, Amazon Cloud, Apache Spark, Microsoft Azure, TensorFlow for machine learning, deep learning, etc. So Bioconductor is tied into a lot of these, either directly or indirectly, or is becoming much more cloud native as time goes on. This is coming back to something that Amy asked about. Bioconductor has a lot of education, training, and a very active community. We have the core infrastructure, which maintains the website, the build packages, etc. But the community contributions here can be at the level of answering questions on a support site, to teaching to underrepresented or under-resourced communities. For example, I taught Bioconductor to a group in Africa, around 100 African genomics and bioinformatics researchers. It's one of the advantages of that COVID has given us, is that kind of thing can happen quite regularly. It's really just a question of one of us taking time to do it. As the project's grown, we've moved away from an informal governance process to a much more formal one. There are 3 governing bodies. There's a technical advisory board. This is this technical advisory board really advises the infrastructure pieces of Bioconductor, the software development aspects, etc. There's a community advisory board. This is relatively new, and the idea here is that as our community has grown, we really need to focus much more on how we have on our impact, and particularly our equitable impact across the research community. So the community advisory board is responsible for lots of efforts, and most of the members, many of the members, have tie-ins to groups that are focused very heavily on diversity, equity, and inclusion, and workforce development, especially in underrepresented or under-resourced communities. And then the Scientific Advisory Board, this is actually one of the longest standing formal bodies. This is a mix of internal and external folks, folks who are deeply involved with Bioconductor and those who are outside of the community, the Bioconductor community. The idea here is to have a high-level view of where science is going, in particular high-throughput biology is going, so that if there are initiatives that Bioconductor needs to put together to change course or to augment current software contributions, we can do that. How can you get involved? A few different ways. This is where, if you have the slides, feel free to jump in with the links here. A relatively new, in the last 4 years or so, contribution to the Bioconductor community is the Slack community. In the upper right-hand corner of that slide, you can see there's 1,700, almost 1,800 active members in the Bioconductor community. Along the left-hand side, you can see some of the rooms that are available. And of course, the Bioconductor Slack is open. So you can feel free to if you join, to make a room or a Slack channel that meets your needs and then invite people to it. It's pretty active, and a lot of the Bioconductor initiatives have Slack channels here that where the main communication happens. Sign up for the support site. One of the things that, if I were spending more time with you all, I'd ask you to do is not only to sign up for the support site, but to either ask or answer a question. And for many folks, asking and answering an online question in an online forum is a terrifying or anxiety-provoking activity, but it's one of the best ways of learning and maintaining a breadth of knowledge, is really just kind of peruse the questions that go into a support site like this, and answer them where you can. Attend a Bioconductor conference. The conference this year is the first time hybrid since COVID started. But again, an advantage, or we've seen opportunities with COVID. And so last year, the Bioconductor conference was into the high triple digits, and we're expecting more like 1,000 participants this year. The costs are very, very low. For the typical attendee, it's $50 if you attend online, and for students, it's $10. We have funding for scholarships, so that if $10 is too much, then students can ask for and will likely receive scholarship support to come for free. And these conferences usually run for 3 days and include a mix of scientific talks highlighting both the scientific application of Bioconductor, but also software packages. And then a chunk of the time is also spent on hands-on workshops, and those are done synchronously online. So opportunity there. And find and attend an event. Bioconductor supports the conferences, but there are lots of other events that are Bioconductor related, and we rely on contributors to tell us about these things. But once they do, those become available through our event catalog. There's also an iCal, a calendar that you can subscribe to, so that you can have this on your Google or Outlook calendar. And then Amy asked earlier about teaching. Another relatively new contribution to Bioconductor is Orchestra. Orchestra allows contributors to develop educational materials or workshops, and then we can encapsulate those into a compute environment that we can then share with participants. What does that look like? You log into the website. That's the left-hand panel. Once you do so, you'll get a list of potential workshops that you can attend or join or use on your own. Underneath there's a little blue button that you can't see. I covered it up, sorry. Launch. And if you click that launch button next to the workshop that's interesting to you, what you'll get is an RStudio instance running in the cloud that will have all the software pre-installed. And the workflow itself, or the tutorial or the workshop itself, will also have materials inside there. So you essentially work either interactively with someone presenting the workshop to you, or if you want to work on it yourself, you can do that as well, just in an environment where you know things will work because we've built it that way. And then finally, for those who are thinking about teaching our Bioconductor, or actually really any computational or data science approaches, we can use Orchestra to support those efforts and allow you to teach really any place in the world. We've had up to around 700 people using Orchestra simultaneously. It runs on cloud-based infrastructure, so essentially there's no impact, and everyone sees a nice a nice environment where they can do their work and follow along. So another quick exercise for those who this doesn't feel right, ignore it, but I'm going to give everybody a minute or two to think about this. This is just really think about either going to the support site, peruse a couple questions, maybe sign up, joining the Bioconductor Slack, click that link, put your email in, you'll join. No risk to that. It doesn't flood you with emails or anything like that for people who haven't used Slack before. It's a really nice environment for communication. Look at the agenda for the conference and potentially register, or find Bioconductor course materials that might interest you. And we have those going back maybe 10, 15 years. So there's a lot of course materials available. And if you navigate to that site, you can find that you can search for stuff. So I'm going to stop for a sip of coffee, let you all think about or navigate to one of those sites. And then maybe we can pause for questions as well.
So I have a question actually from kind of early on. You had mentioned that there are a number of workflows in addition to just the software packages and data sets, et cetera. And so where would attendees find those workflows on the Bioconductor website? And do you have like an example of what some of the workflows entail?
Yeah, let me see if I can find navigate to that for you. Sure. So on the Bioconductor website, there's a link at the top, help. And then if you look at the documentation, there's a workflows bullet point, the second bullet point down. And I will say that the Bioconductor website was developed about 10 or 12 years ago, and it hasn't really changed much. So some of these things that take two or three clicks really should be front and center. But the workflow lists, particularly those associated with gene expression, some of them can be really quite in depth, with a lot of explanation about statistical models, why people make assumptions, or what assumptions are made, etc. So they can be really helpful, not just for learning how to do something, but why to do something. Other questions? Did I answer that okay, Nadia?
Yes, thank you. I'm looking at the workflows now. Thank you.
And I will mention that for many of if you look on the software list, the workflow packages are also listed there. For many of these, well, for all of these workflow packages, they're available in source. The source code is available. So, Nadia, I think you're leading to like, if I wanted to use this myself, could I change it? Yes, you can. And if you have trouble finding those, just let us know.
Great, thank you.
Sure. Any other questions? Well, I'll assume that all 60 people or 65 people who are online are now engaged with the bioconductor community in some way, shape, or form. So we've talked a little bit about analysis software packages. I want to switch gears a little bit and talk a little bit about the data resources that are available through Bioconductor. There's a few different flavors of data available. These are typically available through software packages, but they access data related data resources. Annotation Hub, you can think of Annotation Hub as a one-stop shop for the kinds of information that you would need to understand things like which genes do what, for example, which genes have what function, the relationship between genes in one organism, human and mouse, for example, things that are not necessarily part of the experiment, but are necessary to understand the experiment. That's stuff that lends itself to Annotation Hub. There are 30 plus thousand curated public reference data sets. Those are, Annotation Hub is a software package. You can download it or you can install it. And then there are lots of capabilities for searching and then accessing those curated public reference data sets in the form of bioconductor objects. And remember, we talked about the bioconductor data objects being sort of standardized so that these are reusable and interoperable across Bioconductor. The next sort of big category is Experiment Hub. Experiment Hub has a slightly different kind of data. These are, rather than being annotation data associated with understanding a specific experiment, these are actual experimental data sets. They're user submitted. They're often publicly available data sets that have been curated in some way. And then they have often have associated code and documentation with them. So we'll talk a little bit later about curated TCGA data, curated cancer genome atlas data. Curated metagenomics data is one that I'm involved with now. We're looking at large-scale metagenomic data and its relationship to cancer in particular. Microarray annotation resources, there are lots of those available. And then also we're going to talk about programmatic access to cancer and biological databases. The nice thing about some of these data packages, these data resources, is that for free you get versioning, you get provenance. Where did the data come from? How are they processed? You can find things pretty easily because there's a website that's searchable, etc. If you want to make your data or collaborators' data more useful for marketing purposes, for example, marketing in the sense of people citing your paper, if you make an Experiment Hub, a data package out of your published data, you're exposing that to people, a community of hundreds of thousands of bioinformaticians. People who are, for example, like Nadia, trying to teach something, if there's a data package that includes interesting data, you might use that rather than having to have your students or write code to download and then understand those data. You just grab the Experiment Hub data package. And then interoperability across the project. So these Experiment Hub or Annotation Hub packages, as I mentioned, they're designed for interoperability. This is kind of what Annotation Hub looks like. Essentially, in R, you can ask for R to start up a little browser. This is not public. This is not available on the Bioconductor website. You have to install the Annotation Hub package, and then you can browse locally, do searches, etc. That's the left-hand side. Or on the right-hand side, you can use code to query Annotation Hub and then download those results. Experiment Hub works somewhat similarly. When you install the Experiment Hub package, you're not actually installing all of the data. You're not pulling in all the data. You're just grabbing, you're essentially installing the ability to find metadata, to find the data themselves. And then there's a second step to actually get the data. For those who want to, like everything in Bioconductor, there's documentation about how to create an Experiment Hub package. There's also documentation about how to create an Annotation Hub package. These are nice resources for getting another way to get involved with contributions to Bioconductors. So not actually write a software package, but to write an Experiment Hub package. I'm going to switch gears a little bit and start talking about public data resources in Bioconductor. This will be the last few minutes. I'm going to skim through these slides pretty quickly. But Levi Waldron and Hebekans and I have done a Bioconductor workshop as part of the Bioconductor conference for the last few years. And we cover this material in about an hour. It's, again, kind of ridiculous to try to cover all this material in an hour, but we try to do it. And the goal here is to introduce Bioconductor packages for finding, accessing, and using large-scale public data resources, including Gene Expression Omnibus. That's a database of gene expression. It's about 4 million samples now. The Genomic Data Commons, or the GDC, that's NCI's, the National Cancer Institute's repository for genomic data. And then Bioconductor hosted curated resources for metagenomics, pharmacogenomics, and TCGA. If you want to run the workshop yourself, it's available as a package that's installable. And you can also run the workshop using Orchestra, the online platform. Go and search for public data resources. Again, I'm going to go quickly. Hopefully you have the slides, so you can come back to look at these. Curated data resources. Levi Waldron's group, he's at the City University of New York, has spent really a large chunk of his career developing high quality curation efforts around cancer genomic data, and more recently around microbiome data. Here's the process that they were using, that they have used in the past to get those data and then begin the curation process. And there's a paper from back in 2020 in JCO Clinical Cancer Informatics on this process. On the right-hand side of this slide are data resources or are Bioconductor packages, and the relationship between those, the utility of those packages and the ability to use them for integration, data integration, ease of use, and completeness. And so curated TCGA data, CBio portal data are highly integrated. That is, they've curated those data sets so they're connected. The different assays and samples are connected to each other in a very complete way. They're fairly easy to use, but they really rely on high-level data ingestion. So if you need a lower level data or need more details about those data, they may not always be available in the top packages on the list. And then at the bottom is our packages like Genomic Data Commons that allow full access essentially to any data that you could find in NCBI or NCI's GDC repositories. These rely on core data structures. We talked about these sort of standardized data structures. Everything that goes into these curated data repository, curated data efforts is in the form of one of these core data structures, again, meaning that it's highly interoperable with software packages in Bioconductor. This is just for a different audience. We talk about S4 data structures, but in this case, let's just focus on the right-hand side. These are the interrelationships between the data types that are available, the diseases that are covered, and the platforms that are used to do the assays. And you can see it's a pretty complex environment, and this is just TCGA data. Here's an example of how you could use TCGA-omics data through another package called TCGA Biolinks. Curated TCGA data is one of these sort of high-level, very integrated data pack or software packages for accessing TCGA data. Another example. For a different data resource besides TCGA, there's another project, CBioPortal. It's been running for maybe 8-9 years. CBioPortal grabs oncological data sets of various types and integrates those data into a portal that's available for online searching, cbioportal.org. And this multi-omic data is available for, it's probably more like 350 now oncological studies, more than 20 primary sites. And CBioPortal data is a curation effort that, or a software package that allows you to access the data in CBioPortal from directly inside R. Microbiome data is something probably a lot of us need to spend more time thinking about. If you subscribe to any nature science journal, update site that gives you new data or new manuscripts coming out from these journals, it's pretty regularly these days includes microbiome or metagenomic data and the relationship between those, say, gut microbiome and some disease process. Curated metagenomic data has manually curated, uniformly processed metagenomic data. We just updated this from 10,000 samples to more like 21,000 samples. Those are available through Experiment Hub as a data package. And 16S data are also available from the Human Microbiome Project. And the data themselves you can get from these websites, from various portals or websites. But what you won't get is the integration of the data with manually curated sample metadata. For example, what kind of cancer, age of patients, etc. All of that is built into these microbiome data resources. Pharmacogenomics data is another complex data type. Here's a history of data sharing for microbiome or for pharmacogenomic data. A lot of these data resources are available for download. You can go and you can download files. But what Ben's group has done is to develop software packages that allow you to access the data, but also include statistical and machine learning approaches associated with those data, and also the comprehensive metabolic or molecular profiling that go along with the sensitivity or phenotype screening. These are the various data set names that are available, and my guess is that that list has grown since the last time we gave this talk a couple of years ago. Drug sensitivity and drug perturbations are both available. And some of these perturbation data sets are quite large, millions of experiments. Radiogenomics is also a relatively new field with, again, large-scale studies with a lot of the same statistical analysis and machine learning approaches being applicable. For those who are a little bit closer to gene expression data or want to think more about gene expression data, the Gene Expression Omnibus is this large-scale, publicly available repository of gene expression data. And GeoQuery is a package that I wrote back in, I believe it was 2006, 2007, that allows Bioconductor users to access any of those data directly from Bioconductor. And of course, again, you can download the data yourself, but the data can be complex. And if you're downloading things by clicking on links and stuff like that, then that becomes a break in your reproducible research, chain of custody, I guess. So using GeoQuery to grab data that you're going to use or reuse for an analysis can be quite powerful. The kinds of things that you can do with GeoQuery and with other packages like it that we've been discussing, we can make a principal components analysis plot from public data using, for example, 10 lines of code. So this is we're looking at a principal components analysis plot from a data set with a little over 250 samples with 4 different kinds of cancer and also normal tissues. Here's the code to actually do that work. And you can see it is a total of actually 9 lines, I guess, here. And those 9 lines or 10 lines of code will produce this. And if I can share with you that code, then you can reproduce this figure directly. No need to go to the website, no need to download anything, no need to find files. The code fully encapsulates the ability to create this PCA plot. And once you have the code, then you can replace the data set with another cancer data set or a lupus data set, et cetera. And you can produce PCA plots from those as well. Genomic Data Commons, because this is a talk about cancer genomic or cancer data around cancer data science. The Genomic Data Commons package is this is a bridge from the NCI GDC to Bioconductor. And this is not the easiest package to use, but it's the most complete package for accessing the GDC. And I'm going to just kind of skip through slides here, but this is the data model for the GDC, at least as of a year or a couple of years ago. What I mean by data model is that these are the things, those circles are the things that are stored in the GDC, and the lines are the connections or the relationships between the things that are stored. And what the Genomic Data Commons package does is to simplify that data model so that you can write code that is reproducible and reusable without having to understand all of those data model or that entire data model. As an example, we can explore the somatic variants in cutaneous melanoma using Bioconductor tools. And here's what the code looks like to do that. This code will generate what I'm going to show you next. And we're going to show the relationship between the code and the website. Here's what we're going to get as a result. So that code, just those few lines of code can generate this. In the genomic data commons package is essentially going to allow us to write in code what we would do by clicking on the website. So we're going to look at files. Here's what it would look like on the website. We're going to choose cutaneous melanoma. Here's how we choose cutaneous melanoma using the package. We're going to grab somatic mutation data from MUTEC2 in MAF format. Here's how we do that in code. Here's the file that we're going to be accessing. Again, we could click on this, download it, and then load it into R, or we can use this line of code to grab the file and then download it. MAF tools allows us to take, remember we talked about data containers, allows us to take the data that we just loaded from the Genomic Data Commons, and there's a tool called MAF tools that allows us to take that data in that common data container and do all of these other things with it. So we can use MAF tools to read the MAF file, and with one more line we can produce this. We can also, with a couple of extra lines of code, produce these kinds of things. And all of this, because of this high-level abstraction that Bioconductor offers, this software encodes knowledge. I don't have to know how to produce these plots. I just need to find the right package, in this case, MAF tools. And with just a few more lines, I can produce these kinds of plots. I know those last few slides were a bit of a whirlwind, apologies. But like I said, the slides are available, and I hope I've given you enough of an introduction to Bioconductor resources that you can start to explore a little bit on your own. So with that, I'll stop and take any questions, comments.
Yeah, hi, Sean. I can start with a quick question. So you talk about all these tools, and I'm thinking of it because next week we are going to talk about the integration of multi-omics data. So I'm wondering, like, I guess my first question is in terms of data resources, because we always have trouble in finding public available data with both like DNA sequence information and RNA-seq information. So what are the places you think have one individual with both types of omics data that kind of public available, not like DB gap you need to apply for access?
Yeah, that's a great question. We typically think about human data when we talk about these kinds of data integration efforts. And I mean, just by the nature of human data being identifiable, much of the DNA sequence, the sequence level information is going to be protected. That said, TCGA, for example, has, in addition to DNA sequence information, which is protected, has typically many assays, in some cases, multiple assays for the same data type. So for example, it's not uncommon to have both array-based and sequence-based methylation data. But certainly for many TCGA studies and associated and other data sets in the Genomic Data Commons, there's data for RNA-seq, for microarrays, for methylation, for proteomics. And of course, a big part of data integration is integration with the sample level information. So those data are typically available, and the curated TCGA data package might be a good place to look. We didn't talk about it specifically, but there's a data container called multi-assay experiment that incorporates, that allows, that contains in Bioconductor, contains multiple different assays for the same samples. So that data container allows you, for example, to have proteomics and transcriptomic data in the same container. So slicing and dicing that way. In terms of DNA sequence data, there are mouse data that are available that are typically not so clean to get, that you could look into. And the other is looking at cell line data. The cell line data will again, like TCGA, will have multiple assays associated with them, along with variants. Typically, those are typically available as well. But I think, I mean, to get to the crux of the issue, data integration, a lot of data integration is often just about curating the data to begin with, finding the data, and then putting it into a form that allows you to perform the data integration. And I'd love to say that Bioconductor's figured it all out, but a lot of it is really left to the user.
Okay, thank you. And a sort of related question is on the tools for the such kind of a integrative data analysis. So I guess since Bioconductor is so popular, so what are the I don't know, what are the tools available there? Or if you want to search it, like just do you have any suggestions on doing that?
Yeah, let me see if I can find quickly here. Here it is. I'm going to put this in the chat. So Mike Love is an investigator at UNC. He's the developer of the DSeq 2 package, which is one of those top packages in Bioconductor. But he's developed an interest in multiomics, and it turns out that there are lots of different ways of slicing and dicing multi-omic data resources and data analysis approaches. So he's developed a little website that lists these things. And the cool thing about this particular website is that it's in historical context. These have, there's a lot of historical context here, but these multi-omic integration packages are becoming more commonly used, and particularly as we move, the field has started to dabble not just with these linear algebra, or matrix-based approaches, but also started to look at deep learning and transfer learning approaches for multi-omic integration. But this is a good place to start. So.
Okay, great. Thank you. I'll check that out. Yeah. I think early on, like Jessica asked the question about where to find the workflows. So yeah, just paste the link there. I don't know if you want to add anything to that. I assume that there are a lot of interest after Nadia's question on the workflow.
Yeah, Bioconductor enjoys a wealth of documentation available online. So the workflows website, or the workflows link that's in the chat, is the official workflows site. But just going to follow up on Nadia's points about question about where this comes from. There's a ton of documentation, training, workflows, tutorials YouTube videos. I didn't mention that, but Bioconductor has a YouTube channel that has hundreds of videos. There's a ton of information out there, tutorials for you to peruse and use. Finding the high quality ones can be a little bit challenging, but there are even commercial efforts and massively online courses that focus on genomic data science using Bioconductor. So, yeah. There's a question about, can you please share the link for the website? I think I dropped that in the chat. So it's Awesome Multiomics to the anonymous attendee in the chat, not in the Q&A. Cool. Other questions? And to Jane's point, the slides are available. My understanding from Min is that the talk it has been recorded and will be available as well. Am I right?
Yes.
All right. So all the mistakes I made, I can look at later. Advice to people: don't ever watch your own talks. It's horrifying. All right. Well, thank you all very much for your attention, and please feel free to reach out to me by email or Twitter or whatever. I'd love to hear from you, and of course, would love to see you engaged with Bioconductor. So thanks again.
Yeah. Thank you so much, John. And we actually have a lot of thank you for the fabulous talk, and from the chat. Thank you, and I hope to see you next year then.
Hopefully sooner than that, but yeah.
Yeah.
All right. Take care, everyone. Thank you.
Okay. Bye.